that we have, I was talking about the endocrine hormones, okay, which are produced by endocrine cells and they pass in the blood circulation. Because in the beginning I talked about glands that secretes hormones which circulate in the blood. هنا I was specifying إنه not all the endocrine hormones are produced by glands. We have some most of the endocrine hormones are produced by glands. We call them glandular hormones, okay? But we have other hormones that are also endocrine. They pass in the blood circulation, but they are produced by endocrine cells, which are not part of glands, okay? We can have, for example, scattered cells in a certain tissue, but those cells have the ability to secrete hormones. بنقول لهم endocrine cells, okay, like in the intestine, for example. بالintestinal epithelium, we have the epithelial cells, but between them we can have some scattered cells which have the possibility to produce hormones. بنقول لهم endocrine cells, okay? So those are cells that produce endocrine hormones but are not glands. They are not part of a gland. Okay, and also the adipocytes, which are the cells of the adipose tissue, they produce the leptin. This is the hormone that regulates the feeling of hunger and the need to eat or not in the body. Okay, this is an endocrine hormone also. So they have the possibility to produce endocrine hormones, and they are not glands. And also we have some neurons, like in the posterior hypophysis. And as I have explained at the end of the chapter, the neurons in that produce the oxytocin and vasopressin, okay? They produce hormones. So we call those hormones neurohormones because they are produced by neurons, and those hormones are endocrine hormones. So that's what I was explaining here, that we have endocrine hormones that can be produced by glands, we call them glandular hormones, and other endocrine hormones which are not glandular. They can be produced by some neurons or by isolated cells or by the adipose tissue, for example. Okay?
يعني نحن هيدي الصورة بس هو المطلوب منا نعرفهم لأنه هي أصلا مش موجودة عنا بالكتاب.
هي كمان بس general idea for you، يعني كمان للامتحان ما بس نسألكم عن هيك شغلة بالامتحان.
اوكي.
Okay. Now
دكتور نحن حنشرح chapter 5.
نعم. لا هلا we will finish the chapter 3. Next week we will start by the chapter 4. Okay. Now we will talk about the gonadogenesis in this chapter. So we will talk about how the gonads are formed. The gonads in the male and in the ovaries and the testis, how are they formed? So the gonadogenesis. First of all, we'll start by talking about the indifferent gonads, because at the beginning, when the fetus is formed, first of all, the gonads are indifferent between both of the sexes. That's why we call them indifferent gonads. Okay, and in this part, we will detail the phases through which those gonads pass. So the phases that participate to the formation of those gonads, to the indifferent gonads. Then after a while, those indifferent gonads will start to differentiate to give either testicles, like in the males, or ovaries, and then we will talk about the ovarian differentiation. First of all, we will talk about the indifferent gonads. Now, as you know, after the fusion of two gametes, after the fertilization, which is the fusion of a male and female gamete, we will have a production of a zygote that can carry two X chromosomes, one coming from each gamete. Either the zygote can be carrying one X chromosome and one Y chromosome. So here we will talk about a genetic sex. So the genetic sex of the fetus or of the zygote is the sex determined by the type of sexual chromosomes. Okay, if we have two X chromosomes, this means that this fetus is destined to be a female and it has a genetic female sex. If it has an XY, if the cell or the zygote formed or the fetus carries XY chromosomes, this means it has a genetic male sex and it is destined to be a male. Now, during the early development of the gonads, we don't have in the males, in the uh, fetuses carrying XX or XY chromosomes, the gonads are identical at the beginning. We cannot differentiate between them, and there is no difference, in fact, between them. They are the same. That's why we call them indifferent gonads. So the first stage of the gonads formation, in their first stage, they are indifferent or indistinguishable between females and males, and they are called indifferent gonads or bipotential gonads. Bipotential because later they will differentiate to give either a male gonad or a female gonad. So when they are indifferent, they have the potential to differentiate into both of the gonadal types. We call them bipotential gonads. Now, the gonads, when they develop, they have two cell origin, or they originate from two tissues, the mesenchyma and the coelomic epithelium. So we have two tissues in the fetus that are at the origin of the gonadal genesis or development. The mesenchyma, which comes from the intermediate mesoderm, and the other tissue at the origin of the gonads development is the coelomic epithelium. And those both tissues form the indifferent, or they form the gonads. And at this stage, in fact, the first when the gonads are firstly formed, they are called genital ridges. I'll start by to give details about each idea. But do you know what is, when we say coelomic epithelium, do you know what does coelomic means, or coelom? What does a coelom mean?
Cavity.
A cavity, yes. Do you all agree, or do you all know this or not? Okay. So a coelom is a cavity that is in the body, and like we can see here, for example, here we have the abdominal cavity, which is the cavity in our abdomen. Okay, in this cavity we have the stomach, the intestines, the uterus, um, and many organs. Okay, the kidneys, the gonads will be in this cavity. Okay, so the cavity, the coelom is a cavity, and where are the gonads? Do you know where are the gonads? Let's say the ovaries in the females. Where do we find them? We find them in the abdominal cavity, but where? From the back, on the back side, yani behind the intestine. Okay. we will find them on the back near to the kidneys. Okay, يعني behind the intestines we will find the gonads, the ovaries, let's say, in the females, and we will find also the kidneys. Okay, we will go back to this idea. So a coelom is a cavity, and the mesenchyma develops from the intermediate mesoderm. Here, as you know, during the embryogenesis, we have the formation of those three layers: the ectoderm, the mesoderm, and the endoderm. Okay, the mesoderm is the middle layer here, and the mesoderm will be divided into the paraaxial mesoderm, which is near the fragment or near the neural groove. We have the intermediate mesoderm, okay, and we will have the lateral mesoderm, which will give the somatopleur and the splanchnopleur. Now, the gonads will develop from the intermediate region of the mesoderm, okay? It will, so this intermediate region of the mesoderm, here we have the mesoderm. This is the paraaxial mesoderm near to the neural groove, and here the intermediate mesoderm, we have it, and then we have the somatic mesoderm and the lateral or the splanchnic mesoderm that will give this line. Now this intermediate mesoderm will give the mesenchyma, which will be at the origin of the genital ridge or the indifferent gonads formation. Okay, so the indifferent gonads will develop from this region here, which is the intermediate, the mesenchyma of the intermediate mesoderm. We can see here also the different steps of the embryogenesis. We have here, we have the intermediate mesoderm, okay, and here the paraaxial mesoderm, and here we have the lateral mesoderm. At certain moment, we will have the formation of a cavity, as you can see here, the cavities. Okay, we have the cavities formed, and we have also, so this will be the coelom. This cavity is called the coelom, and this part here, this is the intermediate mesoderm. Okay, the intermediate mesoderm will develop here. We'll have the intermediate mesoderm that will give the genital ridges. What does ridge mean? Do you know what does ridge mean? It's like an outgrowth، يعني مثل انتفاخ. okay, so كيف عنا tumor for example تعمل outgrowth، بتكبر وبتنفخ. So at certain moment this mesenchyma here, this part of the intermediate mesoderm will make an outgrowth and it will form a ridge, what we call a ridge. Okay, and so this outgrowth from the inside, يعني كل this pink color inside is the mesenchyme. Okay, is a tissue called the mesenchyme. And look here, we said that we have a coelom. Okay, this coelom, the lining of the coelom is composed from an epithelium. As you know, the epithelial tissue lines the cavities. Okay, so this is a cavity lined by an epithelium. This is a coelom lined by an epithelium. So this is the coelomic epithelium, we call it. So the lining of those ridges here is formed from the coelomic epithelium. Okay, so inside of the ridge is composed from the mesenchymal tissue, from mesenchyma, and the lining of this ridge is composed from coelomic epithelium. Did you understand this?
دكتور فيكي تعيديها؟
Okay. So قولي لي شو ما فهمتي بالتحديد يعني.
هي هيدي فكرة إنه بالجينيتال ريدج يعني اللايننج تاعولها من فوق.
Okay. So بس أول شيء, is it clear إنه genital ridge is formed from the intermediate mesoderm the intermediate mesoderm بيعطينا الميزنكيما. Okay, this mesenchyma comes from the intermediate mesoderm. Okay, so this mesenchyma will do an outgrowth، يعني مثل بيكبر بيعمل proliferation بينفخ. Okay, to form a ridge, a form of a ridge بيطلع عندي. طيب هيدا الريدج from inside we have the mesenchymal cell لأنه من جوا هو mesenchyma, a tissue called the mesenchyma. ف we will have mesenchymal cells from the inside and but is lined by the coelomic epithelium. يعني مثل ما شايفين عنا cavity هون. This cavity is called a coelom. And as you know, the cavities are bordered by epithelial cells. يعني هون in dark pink أو in red إذا بتكون we have an epithelium أو epithelial cells. Okay, so we call it the coelomic epithelium. هيدا الابيثيليوم كله. So ridge, those ridges here, the genital ridges, from the inside they are composed from the mesenchymal cells, from mesenchyma. وهيدا lining يعني الخطية اللي حواليه يعني ال exterior part of it is composed from from the coelomic epithelium. Is it clear or not?
Yes, clear.
يعني مثل عندك ساك لأقول من جوا في شيء ومن برا مثلا قماش هو أو هيك، يعني هيك بدك تتخيلي organ أو something is forming like a sack. من برا بدل ما يكون عندك tissue عنا إنه قماش، يعني عم بحكي إذا ساك حملتيه عندك epithelium, a coelomic epithelium. The same cells composing the coelomic epithelium, they are lining this ridge. From the inside, it is filled with mesenchyma. Okay?
Yes.
So the gonads are formed from the now at this stage we call them the genital ridges. So once they are formed, this first stage of in this first stage of development, they are called genital ridges or indifferent gonads because they are the same in both of the sexes. They are both of them formed from mesenchyma and from epithelial, coelomic epithelium, from the coelomic epithelium. So they are formed from the proliferation of the coelomic epithelium that covers the underlying mesenchyme. So the coelomic epithelium here covers the underlying mesenchyme. The mesenchyme is inside and is surrounded by this epithelium. Okay. And we have three phases that characterizes the gonad formation. First, we have the genital ridge formation. Then we have, so at this stage, as I have just explained, the genital ridge will be formed, but they don't contain yet the germ cells. And at this stage of development, they still don't contain the germ cells. They don't have germ cells. They are only composed from mesenchyma and the coelomic epithelium. Later, we will have a second step in those indifferent gonads formation, which is the germ cell migration. And then we will have a third step, which is the sex cord formation. We will talk about them. Now, like I have just explained, the first step is the genital ridge formation. The first step of the indifferent gonads formation is the genital ridge formation. So the gonads develop as paired bulges of tissue near the middle line at the back of the abdominal cavity next to the developing kidneys. As I have explained, if you have a cavity, you have the abdominal cavity, and where will we find in the adult, where we will find the ovaries? In the abdominal cavity, in this abdominal cavity near the back, ورا يعني حد الظهر. Okay, يعني كأنهم laying on the back. Okay, and near the kidneys. We will find them near to the kidneys. So here in the fetuses also, we will find those genital ridges or indifferent gonads. They will form at the back of the abdominal cavity. At the back of the abdominal cavity, يعني من وراه. Here we have the vertebral column, so this is the back. In the back, we will find the kidneys. Now, we have the mesonephros kidney in the fetus, but this mesonephros kidney will disappear later and will be replaced by the metanephros kidney, which will be the functional kidney and will remain in the other stages of development, okay? But so we have this kidney, the mesonephros kidney. Next to it, next to the kidneys, the genital ridges or the indifferent gonads will form, as you can see here. So as paired bulges, يعني as a pair of outgrowths. مثل انتفاخ من ورا بصير near the middle line at the back of the abdominal cavity. So near the middle of the abdominal cavity, we will have them formed, and they will appear at the 5th week of development. Okay, so here we have also, this is the back. Those are the mesonephros kidney, so the primitive kidney. This is the primitive kidney that will disappear later. And next to those kidneys, on the back side, يعني at the back of the abdominal cavity, this is the abdominal cavity, this is the cavity. On the back of it, we have the kidneys, the mesonephros, and we have those genital ridges. Okay, so those are the future gonads, the genital ridges, which are formed at the back of the abdominal cavity. Is it clear till now?
Yes.
We have one from this side and another one from the other side here. And as you can see, they are near the midline of the back. Okay. I will just explain a little bit. Yes.
ما فهمت كيف يعني paired bulges.
We have a pair, يعني paired. عنا اثنين منهم، واحدة من هالميل وواحدة من هالميل. As you know, the gonads, we have two of them. هيك. They are a pair.
يعني من هول الريدجز مطرح ما كاتبين Gonadal Ridges في عنا اثنين.
Yes, عنا واحدة هون وواحدة هون. Like the gonads in the adults, كم واحدة بيكونوا؟ مش اثنين، واحدة من كل ميلة.
Yes.
that's what we mean. يعني we have one pair of genital ridges. يعني عنا اثنين بينعملوا، واحدة من هالميل وواحدة من هالميلة.
Okay.
So the first form of their development is called genital ridges. So those indifferent gonads, in their first form of development, they are called the genital ridges, and they are similar in both sexes. That's why we call them indifferent gonads or bipotential gonads. They are the same in both sexes. Okay, they are formed from mesenchyma and the coelomic epithelium. Here we can see we have the abdominal cavity, we have the back one, in blue the mesonephros kidney, and just next to this mesonephros kidney we have those two red forms, which are the genital ridges, one here and one from this side. Those are the forming genital ridges, or the form genital ridges. Okay.
دكتور عندي سؤال بس.
Yes.
دكتور هلأ هو بالعادة الأوفريز بيكونوا ورا الكدنيز أو الكدنيز وراهم؟
لا، الكدنيز آخر شي ورا بيكونوا.
أوكي.
يعني الكدنيز وراهم بيكونوا. Now, I will go back to this figure. Here we said we have the bag and the abdominal cavity with the mesonephros kidney and the genital ridges. If you can see here, we have, this is the gut, this is the heart of the embryo, here is the gut. And here you can see we have, this is a sac, a sort of sac called the yolk sac. The yolk sac in the birds and the reptiles has a feeding function. Okay, it contains yolk and it helps them in feeding the birds and the reptiles. But in the humans, it doesn't have a feeding role. The first blood cells are formed in the yolk sac, then they will migrate to the embryo, but then this yolk sac will disappear. Okay, so it will not stay in the developing embryos in humans. Okay, it will disappear earlier in early stages of development. And next to this yolk sac, we have what is called the allantois. Here, the allantois is like a posterior extension of the yolk sac. Okay, and also this allantois, it has a function in other animals in the urine elimination and gas exchanges, but in the humans it doesn't have a role, but it will contribute later to the formation of the umbilical cord and the placenta. Okay, now we go. Yes.
ما سمعت كتير. انت قلت الشيء اليوكساك انه it will disappear.
Yes.
Why?
During the embryogenesis, it doesn't have a role like in other animals, مثل in the birds and reptiles, so it will disappear later during the development.
يعني ما له شيء أهمية.
بس مثل ما كنت عم اقول بالاول انه بيعمل blood cells. هو the first blood cells are formed in the yolk sac. They will migrate to the embryo, and then early in the development بيصير في عنا the liver that will start to produce the blood cells قبل ما يكونوا عنا بالبون مارو. اوكي، سو the liver بعدين ببلش هو يعمل synthesis للبلاد cells، مش يعني ببطل اليوك ساك. بعدين اليوك ساك will I don't know if I have. In fact, it doesn't have an import. يعني it will disappear later. Okay, in the humans.
Okay.
Okay, we have the allantois. Okay, and the yolk sac is here we have the gut. We have the foregut, the midgut, and the hindgut. So the hindgut is the posterior region of the gut. The midgut is the middle region of the gut or the intestine, and the foregut هي اللي most anterior region of it. Now the yolk sac is connected to the gut by a duct that is called the vitelline duct or the yolk duct. Okay. So we have the yolk sac connected to the gut by the yolk duct or the vitelline duct, and we have also the allantois. Now, وين كنا وصلنا نحن؟ we were talking here about the formation of the genital ridges. Those genital ridges are represented here. Okay. You have this figure in your books also, and here you can see this is the mesonephros, the kidney, the mesonephros kidney. This is the back, the mesonephros kidney, and here you have the genital ridges. Okay, in green you have the genital ridges. This line bordering the genital ridges is the coelomic epithelium, and here you have the gut. And here you have the vitelline duct, which is called in your books, they called it the omphalomesenteric duct. Okay, the omphalomesenteric duct is the yolk duct or the vitelline duct. Okay. Now, this bipotential stage lasts until the end of the 6th week of development. So until the end of the 6th week, the gonads or those genital ridges are still bipotentials. They are indifferent. After the 6th week and at the beginning of the 7th week, we will start to have some differences between the females and the males in the gonads. Okay, so this bipotential stage or this indifferent stage will last until the end of the 6th week of development. And as we have said, the genital ridges are formed from the proliferation of the mesenchyme and are covered by a coelomic epithelium. So those genital ridges are formed from the proliferation of the mesenchyme. The mesenchyme proliferated to form an outgrowth, and it is covered by an epithelium, by the coelomic epithelium. Okay, that's why at this stage, as you can see, the gonads, both of the mesenchyme and the coelomic epithelium, both of them, their cells are somatic cells. So the mesenchyme and the coelomic epithelium are referred to as somatic cell lineage of the gonads. In fact, as you know, in the gonads, we should have the gametes later and the germ cells, but the mesenchymal cells and the epithelial cells of the coelom are not germ cells, okay? They are somatic cells. So the somatic cells of the gonads or of the indifferent gonads come from the mesenchyme and from the epithelial tissue of the coelom, from the coelomic epithelium. That's why we refer to the mesenchyme and to the coelomic epithelium as the somatic lineage of the gonads. So all the somatic cells in the gonads will have for origin the mesenchyme and the coelomic epithelium. Okay. Later on, we will have the germ cells. The germ cells don't have as origin the mesenchyme, neither the coelomic epithelium. We will see from where they will come. In fact, they will come from outside of those gonads. They will migrate to get settled in those genital ridges, but first of all, they don't have for origin the mesenchyme, neither the coelomic epithelium. The mesenchyme and the coelomic epithelium, they are at the origin of the somatic cells in the gonads, and they are referred to as the somatic cell lineage of the gonads. Okay, so they are at the origin of the somatic cells in the gonads. Now, in the second step, we said that the first step during the gonadogenesis is the genital ridge formation. Or during the indifferent gonads formation, the first step is the genital ridge formation, and the second step is the germ cell migration. So we talked about the genital ridge formation. Now we will see how the germ cells will appear and will migrate to the gonads, to those indifferent gonads. As you know, the germ cells, they are the cells that will give the gametes, okay, which are germ cells also. So the germ cells, they are the sex cells. And the primordial germ cells, they are the most primitive germ cells, or the most stem germ cells at the origin of the gametes. So we call them the primordial germ cells, or PGC. Those PGC have an extragonadal localization. In fact, first of all, they are not localized in the genital ridges in the indifferent gonads, and they do not born inside of the genital ridges. They come from outside of those gonads, so they have a different origin. They have extragonadal localization, then they will migrate to the gonads. So first of all, they have an extra they are located outside of the gonads, and then they will migrate to the gonads. So, and not all they have for origin extragonadal origin, but also an extra embryonic origin. So this is the forming embryo. They don't come from any tissue inside of the embryo. In fact, they have an extra embryonic origin. They originate from the yolk sac, from the epithelium of the yolk sac. Okay, so from where do they come? First of all, they identified them, or those primordial germ cells are identified in the epithelium of the yolk sac. So not anywhere in the yolk sac, also in the epithelium near the allantois. Okay, near this region, near the allantois, they are identified here. They are born here in this region, and then they will migrate inside of the embryo and inside of the genital ridges. Okay. So those PGC, those primordial germ cells, are first identified in the epithelium of the yolk sac near the allantois, not in any region of the yolk sac, in this in the epithelial region near of the allantois, and then they will migrate. Okay, they will gather at the level of the allantois and will migrate inside of the embryo. Now, how can they migrate inside of the embryo, and how can they reach the gonads? As we have said, that the yolk sac is connected to the gut by the vitelline duct. Okay, so to get inside of the embryo, they will migrate from the yolk sac, from the epithelium of the yolk sac, through the vitelline duct to reach the hindgut. They will reach the hindgut, and then so here they will migrate from the yolk sac. It is not represented here. Through the vitelline or the yolk duct, they will reach the hindgut. This is the gut, and then from the gut they should reach the gonads. But how they will reach the gonads? In fact, in the abdominal cavity, we have a tissue called the mesentery. Do you know what is the mesentery? What is this tissue? It is a thin tissue that connect or that bind all the organs in the abdominal cavity to the back. Okay, it keeps them attached to the back of the abdominal cavity, and also it connects the organs to each other. And it is vascularized also. It is a vascularized tissue. So this mesentery here, it connects the gut to the abdominal cavity and to the kidneys and to the other organs. So the primordial germ cells, when they reach the gut, they will move to the mesentery, and then from the mesentery, they will reach the genital ridges. They will reach the genital ridges, and they can enter inside of those indifferent gonads. Okay, so they migrate through the intestine from the yolk sac to the yolk duct to the intestine, and then from the intestine they ascend the mesentery because they reach the hindgut, the posterior part of the intestine. So they will ascend the mesentery to reach the gonads. Okay. During the migration, now while they are migrating, they proliferate. They will be proliferating, and when they reach also the genital ridges, they will be dividing. So they will proliferate also after they reach the genital ridge. So while they are migrating, they divide, they proliferate, and they keep on dividing also when they reach or they arrive in the genital ridges. But some of them will die during also the migration, okay, and in the genital ridges. So not all of them will keep on proliferating. Some of them will undergo degeneration and apoptosis, and the other part will keep on proliferating. Here we have another representation. We have an embryo, a fetus, with this the yolk sac, and this is the allantois. And here we have the primordial germ cells. This is the gut, okay? And this is the mesentery. This is the gut. This is a cross section here. We have the gut, and the mesentery here. And here we have the gonads, So they will migrate from the yolk sac near the allantois to via the vitelline duct to reach the gut. They will reach the gut, and then they will migrate through the mesentery to reach the gonads. And all of those blue cells, they are the primordial germ cells. So we can see them. They migrate and enter inside of those genital ridges. Okay, is it clear till now?
دكتور هول primordial germ cells بيكونوا باليوك sac؟
Yes, first of all, they were identified as having the yolk sac as origin. Yani they are born in the yolk sac and they will migrate to the gonads. Okay?
Yeah.
So they don't have a gonadal origin or an embryonic origin. That's why we say they have an extra, the PGC have an extra embryonic origin. They are born outside of the embryo in the yolk sac, and they have an extra gonadal origin. Now, the third step of the product of the gonads, indifferent gonads formation is the sex cord formation. Now, while those primordial germ cells are migrating and are migrating to reach the genital ridges, at the same time, we said that the genital ridges are composed from mesenchyma lined by the coelomic epithelium, okay? So at the same time, where we have this migration of the PGC, at this time also, the epithelium of the coelom, the coelomic epithelium of the genital ridges, the coelomic epithelium lining the genital ridges will proliferate to form ingrowths, okay? Ingrowths inside of the genital ridge. يعني بيعمل proliferation, and those proliferate cells or part of tissues will enter inside of the genital ridges, okay? And those group of cells from the coelomic epithelium entering inside of the indifferent gonads, we call them the sex cords. Okay, so here those cords in so those elongations like in dark red, they are the sex cord coming from the proliferation of the coelomic epithelium. Okay, we have this coelomic epithelium that will proliferate to give ingrowths inside of the genital ridges, and we call them those ingrowths or proliferated parts of the epithelium. We call them the sex cords. They are like cords inside of the genital ridges. So at about the same time as the PGC are entering the genital ridges, a group of cells from the coelomic epithelium will move. Now, while they are proliferating also, and they will move and surround the PGC inside of the genital ridges. Okay, and that's how they form the sex cord, and those sex cords will be irrigated by the blood vessels, so they will be vascularized. Now the sex cord derives from the coelomic epithelium. They are proliferations of the coelomic epithelium surrounding the genital ridges, and the coelomic epithelium proliferates and move to surround the PGC. Now, as we have said, while this coelomic epithelium is proliferating and entering inside of the genital ridges, they will move, so those cords or those proliferating parts of the epithelium will move and will surround the germ cells inside of the genital ridges, and those invaginations or ingrowths of the coelomic epithelium are called the sex cords. Is it clear?
Yes, clear.
Now, until the end of the 6th week, the gonads has the same morphological aspect in both sexes, as we have said. Until the end of the 6th week, the gonads are still the same for both sexes. They have the same morphology and they are composed from the same types of tissue, of cells and tissue. They have both of them, they are composed from mesenchyma, lined by coelomic epithelium. Both of them have sex cords, and in both of them we had a migration of the primordial germ cells. Okay, so the sex cords and PGC are present in both cortical and medullary region of the future gonads. So in those genital ridges, before in those indifferent gonads, as we have said, we have formation of those sex cords. Now those sex cords will be formed in the cortical region of the bipotential gonad, which means cortical means at the periphery. So they are at the periphery of those genital ridges, and also they extend to the middle. We call them the medullary region. We call it the medullary region. يعني middle هي الميدالري region. So in the bipotential gonads, the sex cords are present in the cortical, يعني at the surface, at the periphery of the genital ridge, and in the medullary region, they will extend to the medullary region of those bipotential gonads. So at this stage, until the end of the 6th week, we have the same genital ridges. We have the same gonads for both sexes. We have they are indifferent gonads, having both of them the sex cords at the cortical region and in the medullary region, because as we are going to see later, in the males the sex cords will disappear. We will not have any more sex cords in the cortical region. We will have only sex cords in the medullary region, and in the females, the medullary region or the medullary sex cords will disappear, and we will have only sex cords at the cortical region. Okay, but in the indifferent gonads, sex cords are present in the cortical and in the medullary region. So this is regarding the indifferent gonads. Also, we will see that the genital ducts, once the genital ducts, as you know, in the female, we have the fallopian ducts or the oviducts, and in the male, we have the vas deferens. And so those ducts are not yet formed at this stage of development. Until the 7th week, all the ducts, they are the same in both sexes, okay? And both sexes, they have two types of ducts, in fact. Until the 7th week, the genital tract shows two pair of ducts having the same appearance independently of the genetic sex. So we have two types of ducts. the green here, the green ducts are called the Wolffian ducts, and the ducts in yellow are called the Mullerian ducts, okay? Later on, the Wolffian ducts will be maintained in the males and will be at the origin of the epididymis and the vas deferens, and will disappear in the females. And the yellow ducts, which are the Mullerian ducts, will be maintained in the females and will disappear in the males, okay? But until the 7th week of development, we have the same genital ducts for both both sexes, which are the Wolffian duct and the Mullerian duct. The Wolffian duct is called also the Mesonephric duct. In fact, there are two ducts, a pair of ducts, so they are the mesonephric ducts, and they are derived from the mesonephros, from the primitive kidney, the mesonephros. So this is the mesonephros kidney. It will those Wolffian ducts, they derive from this mesonephros kidney, from this primitive kidney, okay? And we have also the second type of duct, which are called the Mullerian ducts. We have one pair also of Mullerian duct, and they are called the paramesonephrotic ducts. They are localized near to the mesonephros. They are called paramesonephrotic ducts and exist separately from the mesonephros. So they are not connected to this kidney, but they are adjacent to it. Now we will move to talk about the differentiation stages. Here we have the gonads that will start to differentiate into either testicles or ovaries. We'll start by talking about the testicular differentiation. During the gonadal differentiation, as we have said, first of all, we have indifferent gonads. Those gonads, if the embryo has male genetic sex, so if it carries an XY chromosomes, those indifferent gonads will differentiate into testes, and the differentiation in the male starts at the 7th week of development. However, if this fetus has a genetic sex of 46 chromosomes and having XX chromosomes, so if it has XX chromosome, if it has a female genetic sex, the gonads will develop into ovaries, and the differentiation of the gonads in the female will start from the 8th week of pregnancy. Okay, in the male, it will start from the 7th week, and in the females, this ovary formation or differentiation into ovaries will start from the 8th week of pregnancy. So what makes those gonads differentiate into either testes, either ovaries? In fact, we have, as you know, we have some genes at the products of the genes are some genes are responsible for this differentiation into either gonads, either ovaries. However, we have one key gene, which is responsible for this differentiation, and this gene is called the Sry gene. This gene is carried on the Y chromosome, and it is called the sex determining region on Y. Okay, so whenever a person have a Y chromosome, it will have the Sry gene, this Sry gene, and the presence of this gene is determinant for the development of the gonads into testis. If an embryo doesn't have a Y chromosome, so if it carries two X chromosome, it will not have a Y, this Sry gene. And due to the absence of this Sry gene, we will not have development of the gonads into testis. And due to the absence of these genes, the gonads will develop into ovaries, okay? But however, if this if you have a Y chromosome, Y chromosome and an SRY gene is sufficient to induce the development of the gonads into testis. The absence of this SRY gene will lead to the development of the gonads into ovaries. So this is a key gene that determines the development of the gonads into testis. Its absence will lead to the development of the gonads into ovaries. Okay, so this gene is called the sex determining region on Y because it will determine the sex, or yes, it will lead to the development of the gonads and the genital organs. Okay, so the key to sexual dimorphism is the Y chromosome. So the Y chromosome will lead to this sexual dimorphism, will lead to the sexual development into either females, either males. The presence of the Y chromosomes will lead to the development of the gonads into male gonads, and its absence will lead to the development into female gonads. And how these genes will lead to the development of the testis. In fact, it codes for a protein called the TDF protein, which is the testis determining factor. So this gene, after its transcription, it will give a protein called the TDF for testis determining factor, a protein that by acting on the gonads, by being expressed in the gonads, it will lead to their differentiation into testicles. So in its absence, the female development is established. Okay. If primordial germ cells and gonadal cells have an XY chromosome, complement of sex chromosome, we will have the bipotential gonads will develop into testis. If we don't have a Y chromosome, we will have the bipotential gonads that will develop into ovaries. Now, what are the steps in the development of the male gonads or of those testes? The first step of the of the male gonads development is the testicular differentiation. The first step in the testicular differentiation is the formation of the seminiferous sex cords. So we talked about the sex cords. Okay, those sex cords we said that the sex cords surrounded the primordial germ cells, as you remember. We talked here that the formed sex cords will come and surround when they are formed and will surround the primordial germ cells. Okay. Now those those sex cords will become the seminiferous tubules of the testis. As you can see here, we have under the action of this TDF protein, the product of the SRY gene, we have those sex cords in the medullary region. In the males, they are kept or preserved in the medullary region, and they will develop to give the seminiferous tubules of the testis. If you remember, here we have a longitudinal section of the testis. Inside of the testicles, we have the seminiferous tubules where the gametogenesis will take place. Okay, so those are the seminiferous tubules, and those sex cords, the sex cords will be at the origin, will differentiate to give those seminiferous tubules. But first of all, they will not be hollow from the inside. Okay, so they will not, we don't have an empty space from the inside, and later on they will become hollow from the inside. Okay, so we will have development of those seminiferous tubules, and inside of them we have the primordial germ cells, which will give the spermatogonial stem cells. Okay, and we will have also the Sertoli cells. So inside of those cords that will become the seminiferous tubules of the testis, we will have the spermatogonia, or the stem cells at the origin of the spermatogonia, and we will have the somatic cells. As you know, the spermatogonial stem cells will be derived from the PGCs, so the PGC will differentiate into spermatogonial stem cells, and in the seminiferous tubules, we will have the formation of the Sertoli cells, which are somatic cells and are not germ cells. Okay, so the Sertoli cells will develop inside of those sex cords, inside of those seminiferous sex cords, and we will have also the spermatogonia, or the spermatogonial stem cells, that will develop inside of those seminiferous sex cord. At this stage of development, they are without a lumen, but later on they will have a lumen inside of them. So the differentiation of those cells inside of the seminiferous sex cords is the first step of testicular organogenesis. Is the first step during the testis differentiation. Okay, is the differentiation of the PGC into spermatogonial stem cells and the appearance of the Sertoli cells, which are somatic cells. Now, the second step is the appearance of the Leydig cells. The Leydig cells will differentiate. I don't know if you remember if you know the structure of the testis. We have the seminiferous tubules, and between the seminiferous tubules, we will find the Leydig cells. Like here, inside we have the seminiferous tubules, but between them, between those seminiferous tubules, we will find the Leydig cells. Okay, so the Leydig cells will appear in the interstitial mesenchyme between the cords. Here you have the cords that will give the seminiferous tubules in blue. Between them, we have this pink color. This is the mesenchyme, as you remember. We have mesenchyme inside. Those mesenchymal cells will give the Leydig cells, will be at the origin of the Leydig cells, will differentiate to give the Leydig cells. So the second step in the testicular differentiation is the appearance of those Leydig cells, and they have for origin the mesenchymal cells. Okay, they don't have they are not generated from the PGC, for example, and they are not generated from the sex cords, like in the case of the Sertoli cells. No, the Leydig cells appear in the interstitial mesenchyme, so they have for origin the mesenchyma. And they are situated between those cords. We will find them between those cords. Okay. And the Leydig cells are important because they are the cells that secrete testosterone in the testis. Okay, they are the cells at the origin of the testosterone. Okay, and the production of the testosterone will stimulate the testicular development and the development of the Wolffian ducts into epididymis, seminal vesicle, and vas deferens. So it is the appearance of the Leydig cells is very important due to the production of testosterone, which will lead to the testicular development and the development of the epididymis, the seminal vesicle, and the vas deferens from the Wolffian ducts. Now, what happens, also we are still talking about the testicular differentiation, but what happens to the ducts in the males during this testicular differentiation? Now, as we have said, we had two types of ducts, the Wolffian ducts in green and the Mullerian ducts in yellow. Under the action of testosterone, the Wolffian ducts will be maintained. Okay, and they will differentiate to give the epididymis. The epididymis is this upper part above the testis. So it will give the epididymis and the vas deferens and the seminal vesicule. Okay, so those Wolffian ducts will differentiate under the action of testosterone to give the epididymis, the vas deferens, this tube, the vas deferens, and the seminal vesicule. And since the Sertoli cells were formed, okay, the Sertoli cells inside, we said we have Sertoli cells inside of the seminiferous tubules. Inside of the seminiferous tubules, we have the spermatogonial stem cells at this stage of development, and we have the Sertoli cells. The Sertoli cells will produce a hormone called the AMH, the Antimularian Hormone, okay, or which is also called the MIF, the Mullerian inhibitory factor. So this AMH is a glycoprotein hormone. It is produced and induce the regression or the degeneration of those Mullerian ducts. Okay, so under the action of AMH produced by the Sertoli cells, those Mullerian ducts will disappear. Because those Mullerian ducts will give later on the oviduct and will be at the origin of the fallopian tubes, the oviduct, the uterus, the cervix, and the proximal vagina. If we have a problem in the secretion of the AMH by the Sertoli cells, there will be a problem because those Mullerian ducts will be maintained and they will develop to give female genitalia also, okay? That's why it is important to have Sertoli cells, the Sertoli cells well developed, and it is important to have this AMH hormone produced by the Sertoli cells because this will lead to the degeneration or regression of the Mullerian ducts in the males and will lead to the correct development of the male genitalia in the males. If this Mullerian duct is not degenerated, the person carrying the male will carry also female genital organs. Okay. Now, the last step during the male gonadogenesis is the descent of testis. As we have said, the gonads are formed as genital ridges in the back of the abdominal cavity. They are formed in the back of the abdominal cavity in both sexes. But as you know, the testes are not situated in the back of the abdominal cavity in adults or in the children. They are situated in the scrotum. So, but first of all, they are formed in the abdominal cavity, and then they should descend from the abdominal cavity to be localized in the scrotum. Okay, that's why we are talking about the descent of testis. Okay, between the 7th and the 8th months of pregnancy, the fetus should have a descent of testis from the abdominal position into the scrotum. You know what is the scrotum? The scrotum is this pouch adjacent to the penis. Okay, here we have the penis and we have the scrotum, which is the pouch inside of which we will have or a male will have the testis. Okay, so the testis, first of all, they are in the abdominal cavity and they should descend to reach the scrotum, this pouch adjacent to the penis. And in fact, we will have now, not all the details are required, but for their descent, they will descend through an inguinal canal. There is a canal called the Inguinal Canal. Okay, it's a sort of split in the muscles. Okay. Through which the testis will descend to reach the scrotum, and we will have here a ligament called the Gubernaculum Ligament and another ligament the suspensory ligament. Those two ligaments, under the action of testosterone, this gubernaculum ligament will shortens, and because it will shortens, as you can see, the testes are attached to this ligament. It will shorten under the action of testosterone, and once it will shorten, it will pull the testis down, and so the testis can descend through the inguinal canal to reach the scrotum. Okay, they will descend to reach the scrotum. So here we will have the descent of the testis, and this descent is important because if they are not descended, if they remain in the back of the abdominal cavity, this will create fertility problems. In fact, the testis should be in the scrotum because the temperature of the scrotum is 2 to 4 degrees less than that of the body. Okay, and if the temperature is 37 degrees, it's so the gametogenesis needs a lower temperature than 37 degrees to be achieved. If the testis will remain in the abdominal cavity, this will produce sterility in the males. However, if the males who are born without the descent of testis, this case or this situation can be corrected by surgery. Okay. However, if it is not corrected, and if the testis will remain in the abdominal cavity, and if this situation if not corrected, this will lead to sterility in the males. Now, if the testes fail to descend, when they don't descend, this condition is called Kryptorchidism, okay, which is the absence of the testicular descendants, and the testes are called in this case cryptorchid testes. So when the testes are not descended in the scrotum, we call them cryptorchid testes, and this condition is called the Kryptorchidism, which means the absence of testicular descent, but this situation can be corrected by surgery. If the Kryptorchidism stops spermatogenesis, that's why it should be corrected by surgery. If not, it will lead to sterility. So as we can see here, we have during development, we have XY zygote, fusion of an X gamete with a Y, with a sperm carrying a Y chromosome. We will have a zygote carrying an XY chromosomes, and due to the presence of the Sry gene of the Y chromosome, this will lead to the differentiation of the gonads into testes. Okay. Now the Sertoli cells in the testis will produce the MIF or the AMH hormone, the Antimularian hormone, which will lead to the regression of the paramesonephrotic ducts, which is the Mullerian duct. Okay, so the presence of the MIF or AMH will lead to the regression of the Mullerian ducts. However, the secretion of testosterone by the Leydig cells is important for the maintenance of the Wolffian ducts and its differentiation into the vas deferens or the efferent ducts, the epididymis, the seminal vesicle, and the ejaculatory duct. Now, part of this testosterone will be transformed into DHT, which is another androgen, and this DHT will lead to the development of the external genitalia in the males. It will lead to the development of the penis, of the scrotum, and of the urethra, and it will lead also to the development of the prostate gland. Okay, so that's how we can see the gonadogenesis in the males, and that's how the hormones, the different hormones produced in the gonads, regulate the differentiation and the appearance of the male genital organs. Okay. Now I will talk about I will move to talk about the ovarian differentiation. During the ovarian differentiation, as you remember, we had bipotential gonads having bipotential gonads having sex cords. The sex cords in the females will remain will be cortical. However, the medullary sex cords in the females will degenerate at the opposite of the male. In the male here, we have seen that the medullary sex cords will be maintained and will become the seminiferous tubules. In the female, in the absence of the Sry gene and of its protein, the TDF protein, the medullary cords will regress and only the cortical cords will be maintained. Okay, so in the female, we will have the cortical sex cords that will be maintained, and those cortical sex cords, we will call them the ovarian cords. Okay, they will be called the ovarian cords. As you know, we will have the PGC surrounded by those ovarian cords or by those cortical sex cords. They will be surrounded by those ovarian cords. Now, the PGC, they were dividing. We said that the primordial germ cells are dividing inside of the bipotential gonads. They are proliferating, so they will be dividing by mitosis. Now, once they are surrounded by those ovarian cords, they will enter the meiosis, okay? And the primordial germ cell, the oogonia, if you remember, we said we had oogonia that will enter the meiosis, and once they enter the meiosis, we call them primary oocytes, okay? So those PGC will enter the meiosis and will become primary oocytes. However, this meiosis will be blocked at the prophase 1, at the diprotein of the prophase 1. And then those cords surrounding the primordial germ cells. So you have the primordial germ cells in red and the ovarian cords in blue. So those ovarian cords will undergo fragmentation, will fragment to give cells, separated cells, and those cells will surround those primordial germ cells. Here you have an oocyte, sorry, now we are talking about primary oocyte. We have an oocyte, and those fragments of sex cords will come and will surround those oocytes to form follicles, okay? To form what we call a follicle and a primordial follicle. So this first stage of follicular development, we call them, the follicles are called primordial follicles. Okay, so we will have fragmentation of the cords that will give fragmentation of the cords, which will surround the primary oocyte to give primordial follicle. Now, this fragmentation of the cords occurs at the beginning of the folliculogenesis. Here we are talking about a folliculogenesis, which is the production of follicles, because now the germ cells will be surrounded by follicles, and we call this by the follicular cells, yes, by follicular cells, and we call this though we call them follicles. So the primordial follicle are formed by the association of somatic cells, which are the granulosa cells or the follicular cells coming from the fragmentation of the sex cords. Here we have the fragmentation of the sex cords. So the sex cords, you know, they are somatic cells having for origin the epithelial cortex. Those sex cords will fragment to give the granulosa cells, okay? Now in blue, or the precursors of the granulosa cells. Okay, so we call them also the follicular cells. So those follicular cells will surround those germ cells, the primary oocyte, and this is a follicle formed. And we will have also thecal cell formed in the mesenchyma, and the thecal cells will differentiate later and will come from the mesenchyma around the growing follicles. Okay. Now the primordial follicle and the thecal cells are important for the hormonal development. Okay, secretion of hormones. Now also we will have the differentiation of the genital ducts in the females. As we have said during the stage of bipotential gonads, we had bipotential also ducts. We had Wolffian ducts and Mullerian ducts in both sexes, but now in the absence of an Sry gene, in the absence of the TDF protein, which is the product of the Sry gene, we will not have testosterone production. And since we don't have testosterone production, the Wolffian ducts will regress. Okay, so the regression, we will have a regression of the Wolffian ducts. And since we don't have Sertoli cells, we don't have AMH production. In the absence of AMH, the Mullerian ducts will be maintained and they will develop to give the female internal genitalia. So they will develop to give the fallopian tubes, the uterus, the cervix, and the proximal vagina. The proximal vagina is the upper part of the vagina. Okay. So those ducts or those genital organs originate from the Mullerian ducts, which are maintained in the female due to the absence of the AMH, because we don't have Sertoli cells, we don't have AMH, and then the Mullerian ducts are maintained in the females. Is it clear till now?
Yes, clear.
Okay. Now we'll just see again the ducts development in the females and males. So first of all, we have Wolffian and Mullerian ducts. In the males, the Wolffian ducts will be maintained under the action of testosterone produced by the Leydig cells. And since in the males they have developed Sertoli cells, the Sertoli cells will produce the AMH, the Antimularian Hormone, which will destroy, this hormone will lead to the destruction of the Mullerian ducts. So the males will maintain the Wolffian ducts and the Mullerian ducts will disappear or will regress in the males. Okay, in the females, we will have, since there is no Sry gene, we didn't have testicular development, there is no testosterone. So in the absence of testosterone, the Wolffian ducts will disappear and the Mullerian ducts will be maintained, okay, and will be developed to give the internal genital organs of the female, the oviducts, the uterus, the upper part of the vagina, the cervix, okay. And this is a summary of the gonadal differentiation. So first of all, the intermediate mesoderm will give the genital ridges, which are also surrounded by the coelomic epithelium. The genital ridges are the first form of gonads, and they are called the indifferent gonads. They are the same in both sexes, the female and the male sexes. But if the fetus is carrying a Y chromosome, so if it is a genetically male, if it has a genetic male sex with the presence of a Y chromosome, we will have the under the indifferent gonads, in the male genetic sexes. So in the males, since they are carrying a Y chromosome, we will have the SRY gene on the Y chromosome, and this gene will lead to the testicular differentiation, to the production of the Sertoli cells, to the differentiation of the testis, of the seminiferous tubules, the appearance of Sertoli cells. Okay, this will lead to the differentiation of the Sertoli cells, which will produce the AMH, and this will lead to the regression of the Mullerian ducts, and also the differentiation of the testicles under the action of the SRY gene will lead to the differentiation of the Leydig cells from the mesenchyme, from the mesenchyma. So we will have the appearance of the Leydig cells, which will produce testosterone, and under the action of testosterone, the Wolffian ducts will be maintained, and which will lead to the differentiation of the male genital tract, of the epididymis, the vas deferens, the seminal vesicles, and some of the testosterone produced will be converted to dihydrotestosterone, which is another androgen necessary for the development of the external genitalia in the male, like the penis and the scrotum, and is also important for the development of the urethra and of the prostate gland. Now, in the females, in the embryos carrying XX chromosome, and in the absence of a Y chromosome, and in the absence of so they will not have an SRY gene, and this will lead to maintenance. This will lead to the differentiation of the gonads into ovaries. We'll have granulosa, the production, the formation of the granulosa cells and the theca cells, and this will lead to the activation of estrogen production. And in the absence of testosterone, we will have, in the absence of testosterone, yes, the Wolffian ducts will disappear, and due to the action of, due to the absence of Sertoli cells, so due to the absence of this SRY genes, we won't have Sertoli cells, and we won't have AMH, and then the Mullerian ducts will be maintained, and this will lead to their differentiation into a female genital tract. So this is all regarding this chapter. I just like to add something that sometimes we can have genetic problems at the level of the X and Y chromosomes. In some people, during the gametogenesis, sometimes the Y chromosome can lose the SRY gene, and in this case we can have a Y chromosome not so. This will lead to gametes carrying a Y chromosome without this SRY gene. And then if this gamete will fuse with another X, another gamete to give an XY chromosome. So if this this Y chromosome is lacking an SRY gene, the zygote will develop into an embryo and then into a male. In fact, the person will carry XY chromosomes, but due to the absence of an SRY gene, the person will develop female genitalia. Okay, so it will be, it will have a female appearance. It will have like female genital organs due to the absence of an SRY gene. However, this person is genetically a male. It carries XY chromosomes. Okay, but because we have, there was a deletion of this SRY gene, the person will develop into a female. And in the same way, if this gene was translocated into an X chromosome in the gametes of the parents, and then if we had gametes where the X chromosome carries an SRY gene, let's suppose we will have zygotes containing SRY gene, 2 X chromosomes, but we have 1 X chromosome carrying this SRY gene. In this case, it should be a female. it is someone carrying a female genotype. However, it will develop into a male. This person will develop male genital organs, okay? So the females carrying an SRY gene on their X chromosome will develop into to have a male appearance or a male genital organs, and the males carrying the genetic males carrying XY chromosome but who has lost this SRY gene will develop to be like females, okay? They will develop female genital organs. Some of those people will have will be hermaphrodites. It's like they will have mixed genitalia, and some of them will have ambiguous genital organs. And in both cases, they will be sterile. They can't. They have fertility problems, and they are sterile. Okay. Are you still with me?
Yes, دكتور.
Yes.
Okay. Do you have any question?
دكتور
No.
أنا عندي سؤال.
Yes.
بالنسبة للفيجرز الليبلينج بيجينا شي بالإكزام مسؤولين عنهم أو لا؟
No. لا ما في labeling وهيك أشياء. حتى في أشياء مثلا I developed some ideas لأنه هو بالكتاب is very short. This chapter is very short. Okay. أنتو you only have to know what is inside of what's written in your books. But لأنه بشوف شوي إنه it's not enough what's in the books to understand the chapter. I developed a little bit the ideas. بس هو المطلوب منكم بس يلي بالكتاب إنه أهم شيء تفهموا إنه steps تاعوا Genital Ridge Formation. إنه عنا أول شيء sorry أهم شيء تفهموا بالأول the stage of the indifferent gonads, how they are developed. إنه عنا Genital Ridge Formation, and those genital ridges are from the mesenchyme and the coelomic epithelium. Then we have migration of the PGC into the genital ridges, and they migrate from the epithelium of the yolk sac near the allantois to the genital ridges, and they will proliferate while migrating. And when they reach also the genital ridges, and they might die also, but they will keep on proliferating. Then we will have the formation of those sex cords. They are formed from the coelomic epithelium, okay, from the proliferation of the coelomic epithelium. And you should know that regarding the ducts, and the Wolffian ducts and the Mullerian ducts, and the Wolffian ducts are maintained in the males, and the Mullerian ducts will be maintained in the females. During the gonadal differentiation, we have this SRY gene on the Y chromosome, which is determinant for the gonadal differentiation. It will determine if the gonad will differentiate into testis or ovaries. In its presence, the gonad will differentiate into testis. When it is absent, it will differentiate into ovaries. And كمان أشوف شو حاطينكم بالكتاب. هات لنشوف. During the testicular differentiation, we have the production of we have the appearance of the seminiferous sex cords. We have the seminiferous sex cords, which will later be the seminiferous tubules, and they are first without a lumen. Inside of them, we will have the primordial germ cells that will differentiate into spermatogonia, and we will have the Sertoli cells. And outside of those seminiferous sex cords, we will have the development of the Leydig cells, okay? And the Leydig cells will produce testosterone, which will lead to the maintenance of the Wolffian duct, and the Sertoli cells will produce the AMH, which will lead to the regression of the Mullerian ducts. Ovarian differentiation, best you should know that in the absence of the SRY gene, the ovaries will, the gonads will differentiate into ovaries. We will have بالovaries بس عنا الcortical sex cords that will undergo fragmentation, and as a result of the fragmentation of those sex cords we will have the follicular formation. يعني بيعطوني cells at the origin of يعني cells يلي هن ال granulosa cells. Okay, so the fragmentation of those sex cords and the females بتعطينا ال granulosa cells that يلي they will gather with the primordial germ cells to give primordial follicles, and we will have the formation of theca cells also. So this is a summary, and this is what you should know from this chapter. And you should know also that in the male we will have a descent of testis, and if they are not descendant, this case is called the Kryptorchidism. Okay.
دكتور بس سؤال.
Yes.
في بالنسبة قلت شيء إنه تستوستيرون بيساهم بأثر شيء shorten something ما بعرف.
كمان هيدا مش مطلوب، بس عنا للديسنت of testis we have two ligaments يلي هن testis attached بيكونوا they are attached to the testis, the suspensory ligament and the gubernaculum. Here the gubernaculum under the action of testosterone يعني يلي هي produced by the Leydig cells they will shorten بيقصر. okay. هيدا الليجامنت بيقصر، وإذا قصر ما هو attached على التستس يعني بصير كأنه عم بشدهم لتحت، كل ما يقصر بتصير التستس بدهم ينزلوا معه. هيدا الجوبرناكولم بده يقصر، والسسبنسري ليجامنت هو is attaching the testis من فوق بيطول، هيداك بيطول وهيدا بيقصر، فهيك بيقدروا ينزلوا التستس to reach the scrotum.
Okay, doctor, thank you.
Yes. أهلا. اوكي، بعد حدا عنده سؤال؟
No.
Okay. Thank you and see you next week.
يعطيك العافية.
الله يعافيك.